So what we're going to do today, because we have a lot to cover between now and the end of the lecture on Monday, I'm going to leave these on the board, and I'm not going to walk you through them like I normally do. I'll let you copy them down for yourself. But what I've done here is basically I've divided the two boards into innate immunity on this side, and what we're going to talk about is innate and adaptive immunity. This board is dedicated to innate immunity, it's non-specific response, and this board is dedicated to the adaptive immunity. Now, one of the major differences between the two is that the innate immunity is not specific. It's rapid onset, non-specific, it causes a lot of damage, and it really is just a holding action, giving us time to invoke our adaptive immune response, which is slow but highly targeted and has memory. And we'll walk our way through this, particularly on Monday when we talk about the humoral and cellular branches. But what I want to do, rather than covering all that material on the side boards, I want to summarize it here with a series of boxes. In the first box, the top here, we're simply going to describe this and label this box as immunity. And then immunity can be divided into these two great categories. This is the adaptive immunity. It's sometimes referred to as specific immunity. The other major branch here in this box is innate immunity. Now, the innate immunity is sometimes referred to as nonspecific immunity. Now, one of the hallmarks of the innate immune response is that it's rapid, it's nonspecific, and it can be locally damaging. But its function is basically to provide a kind of holding action. A kind of holding action against the pathogens. We're going to divide innate immunity into two big categories. If you take an immunology course, you'll see there are more subtleties than this, but I'm going to divide it into inflammation, which we're going to talk about as being bacteria. And over here in this box, I'm going to talk about interferon. And interferon we're going to target to something known as viruses. Again, if you go on into another course, you're going to find that there are a whole series of interferons. They're all named by Greek letters. It becomes Greek alphabet soup after a while. And interferon we're going to talk about is the one specifically for a viral response. But again, the interferon to viruses and inflammation to bacteria all are under this rubric of it's fast, it's nonspecific, and it can be locally damaging. The interferon, the source of the interferon is from the source is virally, or I'll just say virus infected cells. Over here on this pathway, inflammation, the source of inflammation, is a number of cell types. The source of inflammation will be mast cells. These release histamine. The source of this inflammation will be local proteins. Some of these proteins will be released from the damaged cells. Some of these proteins will originate from the pathogen itself, but they'll be local proteins. Another major player here is a granulocyte. The granulocyte that we're going to talk about here is a neutrophil. And finally, there's a cell called a monocyte / macrophage. Monocytes are simply immature macrophages. And the macrophages are going to play an important role here. And one of the things they're going to do is they're now going to interact with the adaptive immune response. And the adaptive immune response is going to be divided into these two boxes. This is the so-called humoral immune response. The cell type that's responsible for the humoral immune response here is called the B lymphocyte. And the B lymphocyte secretes antibodies. And these antibodies bind to antigens in the extracellular environment. So these B lymphocytes are going to be secretory cells. We'll talk in some detail where they come from and how they're selected. But these B lymphocytes are going to secrete antibodies that will bind to antigens to form an antigen-antibody complex. Now, one of the hallmarks I want you to remember, and we'll come back to it more on Monday, is these B lymphocytes are secreting antibodies into the extracellular space. And in the extracellular space, they bind to antigens. The other branch of adaptive immunity, do it this way, the other branch of adaptive immunity here is the cellular branch. The cellular branch is characterized by T lymphocytes, which are subdivided into T helper cells and T killer cells. Now, in contrast to the B lymphocytes secrete antibodies that bind to antigens in the extracellular space. These antibodies are floating free. They're secreted proteins. In contrast, the T lymphocytes, both the T helper cells and T killer cells, contain T cell receptors. Let me put this in separation. They bind to T cell receptors. They contain T cell receptors that bind to antigen fragments. On other cells. We'll see that these antigen fragments are presented on MHC1 and MHC2 proteins. Major histocompatibility class 1 and major histocompatibility class 2 proteins. But that's basically a summary of everything we're going to talk about today and on Monday. Most of what we're going to emphasize now is on this side of the track, the innate immune response. And by the end of it, we'll see that these macrophages provide a kind of bridge between the innate immune response and the specific or adaptive immune response over here. So what I want to do then is tell a short story about interferon. And there are several different kinds of interferon, and they're all labeled by various Greek letters. I'm not going to divide it up, but what I want to do is talk about interferon as a response to viral infection. And the story I want to tell here is one of influenza and epithelial cells. We could tell the story from the point of view of any number of viruses. We could tell the story from the point of view of hepatitis viruses in the liver. We could tell the story from the point of view of macroviruses and Ebola virus. But what we're going to do is we're going to tell the story from the point of view of epithelial cells and influenza viruses, simply because it's something we can all be more or less familiar with. We all are aware that we're coming to the end of the flu season now. We're all aware of how susceptible we are to influenza. If we're in a crowded room and someone sneezes and they are infected with the influenza virus and they have their epithelial cells putting forth these viruses, they'll basically spew viruses all over the place. You breathe them in and you're susceptible then to being invaded by those viruses. So I want to tell you a little bit of the story of the virus and a little bit of the story about how we get a non-specific response. So we're going to imagine here that we have these epithelial cells all lined up. This is classic epithelium. This is going to be respiratory epithelium. These respiratory epithelial cells are just classic epithelia. They sit on a basal lamina. But in the upper respiratory tract particularly, they're exposed to the outside world. So as we talked about the tonsils yesterday, every time you breathe in, you're taking in pathogens from the outside world. Now the pathogen I want to tell you about is one of these virions. And I'll make it a little bigger here, and then we'll talk about it being a little smaller. And this is intended to be an influenza virus. The influenza virus is a remarkably simple creature. It only contains, I think it's 11 genes, and it makes about 14 proteins. And what's more is that it doesn't contain DNA. It's an RNA virus. And so what happens is that these influenza viruses have on their surfaces transmembrane proteins. This is a lipid bilayer. The lipid bilayer has been borrowed, has been stolen from one of the host cells, but this lipid bilayer now is punctuated by these proteins, and one of the major proteins that goes to the outside world is called hemagglutinin. It's 1M. Hemagglutinin is this protein, it's a transmembrane protein in the virus. There's then another transmembrane protein here, I'll put it in blue, called neuraminidase. The neuraminidase is a transmembrane protein that goes to the outside. Now what we're going to see here is the neuraminidase is essential for virus escape. And the hemagglutinin is essential for cell invasion. So two of the major proteins are these transmembrane proteins. There's a third one that's on the outside. The others are mostly cytoplasmic, and they're involved with the machinery that will replicate the RNA and work through the RNA replication system. But all I want to tell you about here is simply that what will happen is there will be transmembrane proteins. I'll put these in purple. There will be transmembrane proteins, which are our very own transmembrane proteins. These are actually hemagglutinin. Hemagglutinin receptors. So what they'll do is they provide a site to which the viral particle will attach. Basically, the viral particle will use its hemagglutinin molecule and bind to that receptor. The binding of the virus to this receptor starts a process in the epithelial cell. It starts an endocytic process where now this viral particle is being taken into the cell. And eventually, once it's inside the vesicle, the viral particle then escapes, goes out into the cytoplasm. And in the cytoplasm, the viral particle uses its genetic machinery to take over the protein synthesis machinery of the cell. Basically, this unsuspecting epithelial cell now becomes a host for the influenza virus. The influenza virus now replicates itself, makes millions and millions and millions of copies, assembles those viral particles, and then the viral particles, once reproduced, now go back up to the surface, fused with the surface, and then by exocytosis, they escape. Now, it turns out that the hemagglutinin protein is necessary for the virus to gain access to the cell. That's the keys in. The neuraminidase, what the neuraminidase does is it digests sugars. These are sugars that are in part of the glycoprotein coat. They are part of the sugars on all of the transmembrane proteins. And so what happens is the neuraminidase now will help dissolve, it will help digest the sugars on the proteins, and that allows for a route out for the viral particles. So in short, what's happening is this unsuspecting cell binds one or maybe two viruses. The viruses then find their way in, and then they co-opt the protein synthesis machinery of the cell to make many hundreds of thousands of copies of their own. And then through the neuraminidase, these guys break out, and now they are in a position to invade neighboring cells. So fundamentally, you get this amplification, cell after cell after cell, each one of these cells generating potentially tens of thousands of viruses that will now infect the neighboring cells. This is the first time, presumably, your immune system has ever seen this virus, so it doesn't have any specific immune response. Now, if you're lucky, you may have had a vaccine. And one of the vaccines, we'll see a little later, is an antibody which is going to cover over these hemagglutinins. It will basically bind to the hemagglutinins, and that will prevent the viral particles from gaining access. That's basically the way vaccines will work. If you get the disease, and you're quick enough about it, within about 24 to 48 hours, you can take neuraminidase inhibitors, something like Tamiflu. And what they do is they block neuraminidase. And because they block the neuraminidase, they prevent the viruses from escaping, so they can reduce the severity of the disease by a whole lot. But for now, what I want to think about is our natural innate response. The natural innate response being interferon. So interferon is a response to the viral infection. What happens is when this cell becomes infected with viral particles, this starts a process inside the cell to synthesize and release a molecule called interferon. This cell's a goner. There's nothing we can do about it. And what it does as part of its death throes, it's releasing this molecule interferon. And this is a clarion call to all of its neighbors. And on all of its neighbors, there will be interferon receptors. So now the interferon will bind to these receptors, and it will be taken up into these cells. And it starts a whole cascade of events. But the long and the short of the cascade of events is the interferon reduces the rate of exocytosis and endocytosis. It upregulates systems to digest RNA. So it effectively reduces RNA availability. In cells. And in part, by reducing the availability of RNA, is it reduces protein synthesis. In effect, what the interferon is doing is it's saying, we have an emergency around here, and what we're going to do is we're going to tell all of these neighboring cells to reduce their protein synthesis levels, their RNA levels, their endocytosis and exocytosis levels. You're basically going to slow down the whole metabolic activity of these neighboring epithelial cells. It's a high price to pay. You're basically making all of these cells not as functional as they used to be. Meanwhile, the interferon is also going out and mobilizing other systems. It's what gives you fever. What makes you achy. It makes you feel terrible. It's largely the interferon. And that's all part and parcel of this non-specific response, basically to slow everything down. To some extent, feeling this terrible is a warning that you should go rest someplace and let your immune system take over. But this is a first response because this is the way we slow down viral replication. We can think of this as a kind of holding action until we can turn on our immune system and turn on a specific response that will begin to recognize parts of these viruses as foreign and attack the viruses and attack the virus-infected cells. We'll come to that idea on Monday. But for now, we're simply looking at this non-specific story of the response. I want to point out one more thing here, because you may have seen this, that these influenza viruses have names. The nomenclature is to have an H and an N. And the nomenclature then has an H and it's a number, like H5N1. This H tells you that it's the hemagglutinin, the N tells you it's the neuraminidase protein. There are something like 11 different known hemagglutinin molecules, and I think it's like 9 different N molecules. And these then will give influenza viruses different characteristics. This one, H5N1, is avian flu. It's rare in humans. There have only been something like 600 cases of H5N1 in humans. But among those 600 cases, something like 400 have died. So it's extremely lethal. And the problem here, the good news right now is that for humans, the receptors, the hemagglutinin receptors for the H5 on H5N1 are not in the upper respiratory tract. They're way down deep in the lung. They're really far away from the upper respiratory tract. And those people who have gotten H5N1 have been working in close proximity to infected birds. They've been living with the birds. They've been slaughtering the birds. So they've been really close to them. And presumably that's because the hemagglutinin on the H5 has gotten down deep into the lungs and bound to those receptors and infected those epithelial cells deep in the lung. The worry here is that there's recombination. The worry here is that this is an avian flu, and most of the flus we know of start in birds. And then they will then interact with usually pigs. So the worry here is when you have a combination of what they call pig poultry or pig duck agriculture. And the worry is you get a sick bird, it infects a pig, and the pig is also sick with another form of influenza, and then there's recombination. And then you get a recombination event where you now get a hemagglutinin type that will bind to receptors in the upper respiratory tract. And it still maintains the viremic characteristics of the H5N1, so it's far more lethal. So this is the worry that the World Health Organization has all the time. They're constantly looking for the new forms of influenza, and they're constantly screening for these different H5N1 combinations to see how lethal they are. Yes?
In this situation, when it comes to like the H5, I guess the H number, N number, is it safe to assume that the lower the number is, the faster it is?
So the question is there a relationship between the numbering system and its lethality or its effectiveness? No. These are simply numbered on the basis of the amino acid sequences and the proteins and the hemagglutinin types. They isolate the different proteins and simply name them as their amino acid sequence. And these guys can mix and match to some extent, but usually they stay one with the other. So the most common, the one that came up a few years ago was H1N1. And that was also, H1N1 was one of the early early flus that came from birds to pigs to humans, and that was the Spanish flu of 1918. And that was an H1N1 as well. And ones with the same numbers can have different levels of potency for reasons I don't understand. I think there's some variation in the amino acid sequence here. And the other horror is that it only takes a few amino acid changes in these hemagglutinins, and you get a wholly different character and a wholly different mutated influenza virus. And because they're RNA viruses, they're kind of sloppy when they replicate. They're not like DNA. And because they're sloppy when they replicate, the possibility for mutations is pretty high. So this is a public health threat. And it's one of these things that eventually we're going to see a pandemic caused by some mutated form of influenza. And that's why the World Health Organization is always on the lookout for this. Yes?
So when we get sick, it's not so much the virus that makes us feel terrible, but our immune response?
The question here is, when we get sick, we feel terrible because of the secondary response to the viruses. That's my understanding. That it's this non-specific immune response that gives you a fever, and it's the interferons which give you this sort of achiness and sort of general malaise. And it's not the virus itself. The virus is working at the level of these epithelial cells. It's the secondary effects of the non-specific immune response that make you feel terrible. Anything else? Yes?
The interferon, is that a family of proteins, or is that like a single protein?
Okay, so the question here is the interferon a family of proteins, or is it a single protein? The interferons are a family of proteins. There are a whole flock of them. The one that I'm mostly referring to here is interferon gamma. But there's a whole Greek alphabet of interferons. And they all do slightly different things. And this is the most general of them, but some of them, but they all have different effects. And they aren't all necessarily relegated to dealing with viruses. Anything else? So the next thing I want to do, this is the interferon story. Now I want to come down through this innate immunity and come over to the idea of inflammation and bacteria. Again, this is going to be one of these rapid, non-specific responses. One of the ideas I was trying to imply here is that the interferon is produced by the infected cell, and that interferon is a non-specific, generalized response that happens quickly. And that this is the first kind of holding action to hold back the pathogenic effects of the viruses until we can move down this branch and get to the specific immune response, which will target the amino acid sequences on the viruses. We're going to follow a similar path here, and we're going to talk about it in terms of inflammation. Now again, inflammation can come in any number of different ways. If you get an allergy, you have a kind of inflammation. But the kind of inflammation I want to tell you about is one where we're going to talk about a bacterial infection just under the skin. This is one we're all familiar with. And we can think about getting one of these splinters under our skin, any kind of response we get, and that's the inflammatory response. And again, what we're seeing is a rapid response to hold the pathogenic bacteria at bay until we can in turn mount a proper specific immune response. So let's spend a little bit of time on the upper board again, talking about inflammation. So inflammation is going to be a generalized response, and it's been recognized for, 2,000 years. There was a guy named Galen, and Galen was a physician, and I think it was the 2nd century AD, and he was a physician working at one of the amphitheaters in what's now Turkey, in Asia Minor. And his patients were gladiators, and so he got a tremendous opportunity to see all manner and forms of inflammation. And he characterized the first description of inflammation. Now let me put this over here, and we're going to come back to it. And these are the so-called cardinal signs of inflammation. And these are redness, swelling, heat, and pain. Because he was part of the Roman Empire, he actually gave these names in Latin. And these names in Latin are still used in medical school, and they have a great ring to them. It's rubor, tumor, calor, dolor. And then once you've gotten to this point where it's red and it's swollen and it's warm and it hurts, then the final stage is you sit there and you complain about it and say, I'm never going to get better, it's going to be horrible, and that's Eeyore. But those cardinal signs of inflammation we're now going to explain in terms of the cellular response. And we can describe all of those responses simply from the effects of the cells and the secreted molecules that affect the vascular system and will then recruit the other set of cells in inflammation. So let's lay out this idea of inflammation and imagine we've driven a splinter underneath our skin. And so we'll start with the classic stratified squamous epithelium. So this is meant to be the epidermis. Those are all keratinocytes. Then there's a basal lamina. And down here in this box is now the dermis. I think it's a little smaller. So this is intended to be the dermis. And you remember that one of the hallmarks of the dermis is that it's well invested with blood vessels. So this is meant to be a capillary. The endothelial cells line the capillary. We've seen this several times before. Now let's imagine we've driven a splinter underneath the skin, and carried on this splinter is a whole bundle of bacteria. So these are intended to be bacteria. Now the bacteria have found a great home. It's warm in here, it's moist, there are plenty of nutrients. So these bacteria begin to replicate and divide. They make copies of themselves. So the bacteria proliferate. And in the process, they now generate a series of bacterial proteins. And some of these are toxic. Meanwhile, in red in here, we have our old friends, the fibrocytes. And then at the interface between the epidermis and the dermis, there'll be some cells I'll put in here in green, and I'm going to label these as mast cells. Mast cells release histamine. And one of the triggers for the release of histamine is these toxic molecules generated by the bacteria. There's also potentially some molecules that will be generated by the damaged fibrocytes. That is, the toxins are damaging the fibrocytes. They're releasing molecules as well, signaling to the mast cells to allow their granules. These are histamine containing vesicles. And these toxins, either the bacterial toxins or the molecules that result from damaging the fibrocytes, signal through a second messenger-mediated pathway for these mast cell vesicles to release histamine. Now, the histamine has targets in a couple of places. The histamine will bind to endothelial cells. And it will also bind to vascular smooth muscle. Now you remember that vascular smooth muscle is going to be someplace upstream here a little ways on one of the arterioles. But when the histamine binds to the smooth muscle, the signal is histamine causes smooth muscle relaxation. So one of the effects of the histamine now is to be released from these mast cells in response to the bacterial proteins as well as the damaged proteins from the cells in here. And that histamine now goes out here and binds to the smooth muscle and causes smooth muscle relaxation. What's that going to do to blood flow into this area? It's going to increase it. What's that going to do to the amount of blood flow in the area? That's redness. It's going to bring more warm blood to it, so that's heat. But it also is going to cause some swelling. The swelling results largely because the histamine binds to the endothelial cells. Where did I put that? They bind to the endothelial cells. So when histamine binds to endothelial cells, it causes them mechanical separation. You can think about it from yesterday as expanding the tiny gaps between adjacent endothelial cells. So if you basically pull these apart a little bit, and you have vasodilation and more fluid moving through here, there's more likely to be fluid escaping across the endothelial barrier. So fluid then is going to come out into the site of bacterial proliferation. This is going to be fluid. That causes the swelling. So now we have redness, swelling, and heat. Pain also occurs because these molecules here will, in some cases, bind directly to nerve endings. In some cases, there are another set of molecules released called bradykinins, they're peptides, which then activate neurons. So there will be sensory neurons activated by the cellular and bacterial proteins. It's probably that these bacterial proteins and the cellular proteins, as a result of damage, are causing the local release from other cells of something called bradykinin, and that causes it to be painful. But that's all local. There's another signal that goes out. The signal that goes out from these bacterial proteins, and also from some of the proteins from the damaged cells, those proteins get into the blood vascular compartment. And they get into the blood vascular compartment, and I need some more room here, so I have to draw a bone. And out here at the ends of the bones, but also in other spongy bone, we have red marrow. And among the cell types that are stored in the red bone marrow, I'll put these in black, are a cell type known as neutrophils. These are a kind of granulocyte. The neutrophils are a type of granulocyte. The granulocytes are all part of the nonspecific or innate immune system. What happens here is these molecules from the bacteria and from the damaged cells will now bind to these neutrophils and tell them to leave home base in the red bone marrow and go out into the blood vascular system. I'm going to blow this up over here a little bit so we can see it in some more detail. One of the effects of the molecules being released by the bacteria, perhaps even the histamines, is to have an effect on the endothelial cells. So these are meant to be the endothelial cells. So these are the endothelial cells here. I showed much smaller over here. The histamines have already expanded the gaps between the two. But they also generate out on the apical surface of these endothelial cells they produce a series of proteins I'm showing here in purple, known as selectins. These are glycoproteins that are basically just kind of sticky. And the neutrophils, too, will have on their surface selectins. And so what happens is that when the neutrophils come into the neighborhood, when the neutrophils are still in the blood vascular compartment, but they're in the neighborhood where these bacteria are proliferating, what will happen is that the selectins on the endothelial cells combined with the selectins on the neutrophils will actually get these neutrophils to stick to the wall. They actually will glom onto the wall. And that also is a signal for these neutrophils to begin flattening out. And they literally crawl between the cells. So what's happening then is the neutrophils that have been mobilized from the red bone marrow are sticking to the endothelial cells, crawling between the endothelial cells, and now arriving here at the site of infection. Now, the neutrophils are pretty nonspecific cells. The neutrophils, now when they reach this site, the neutrophils secrete free electrons. They probably also, along with the free electrons, then will generate hydrogen peroxide, and they generate bleach, sodium hypochlorite. These are pretty toxic molecules. These are pretty corrosive molecules. And what we're doing basically is we're generating a generally oxidative environment out here. The generally oxidative environment is out here to destroy the bacteria. But remember, it's a nonspecific response. It's a holding action. We're just trying to hold at bay these bacteria as long as we can until we can induce a proper immune response. So what we're doing is we're trying to kill the bacteria, but we're doing it in such a way that we're causing a lot of generalized damage. Meanwhile, while this is going on, I can use an orange cell. Meanwhile, back here are other cells in the red bone marrow. These cells are called monocytes. They too are recruited by these molecules released from the bacteria and from the damaged cells. They too then come out of the red bone marrow, get into the blood vascular compartment, find their way to the site of the infection, and they too, because of their selectins, will stick to the wall, crawl across the endothelium, and find themselves here at the site of the battle between the neutrophils and the bacteria. I should say, too, here that these neutrophils don't last very long. They're basically out there to a kind of kamikaze mission to destroy the bacteria. So they're out there effectively secreting all these nasty molecules, and they die too. So you have all these dead cells out here. It's a mess. And the job of these monocytes is then to mature into macrophages. In something like 16 to 24 hours, these monocytes come on the scene and they become macrophages. Remember what macrophages do. They are endocytic cells. So let me go back up to the upper panel to do this. Well, let me do it in the back panel. So the macrophages are now going to have these proteins, and they're going to basically gobble up these remnants of bacteria and basically bacterial proteins. Then they'll bring these molecules into the cell, just as we've seen before with endocytosis. They bring the molecules into the cell. They package them in vesicles. And these vesicles are now destined to go to an old friend. They're headed to a site called the lysosome. These proteins, these large proteins, fragments of bacteria, fragments of the protein, fragments of the bacteria, are now broken up in the lysosome into something known as antigen fragments. These are fragments of the bacterial proteins. And I'll show you these as a kind of blue triangle. So these blue triangles are meant to be the fragments of the bacterial proteins. And then in between, there is another compartment. This compartment is known as an endosome. And in the endosome, there's a transmembrane protein here called major histocompatibility. Major histocompatibility protein major histocompatibility protein 2. We call this MHC2. Well, MHC2 is out here, and when the lysosome fuses with the endosome, it delivers this blue protein fragment that now binds to the MHC2 protein. Then the MHC2 protein in the green vesicle finds its way to the surface, fuses with the plasma membrane, and now this purple MHC2 protein with the antigen fragment attached now goes out on the surface. And so now we have the purple MHC2 protein with the antigen fragment attached to it. And this cell now is a professional antigen presenting cell. And what it's going to do is it's going to leave this site of inflammation and go off into a germinal region in one of the lymphatic tissues. Could be spleen, could be a lymph node, could even be the tonsils or Peyer's patches. Goes out to those sites and will present to those germinal cells these foreign proteins and start the specific immune response. So this is the cell that provides a bridge between the innate and the adaptive immune system. With that, I'll let you go.